Estamos aquí para una conferencia que van a impartir dos profesores tunecinos, Wlaid Hamada, de INAT, que es un instituto que hace enseñanza e investigación a la vez en Túnez, y es especialista en productos bioestimulantes. Entonces nos va a hablar sobre este tema. Y luego va a hablar también Zoubeir Chamekh, que es investigador en el INRAD, viene a ser el instituto, algo parecido a nosotros, pero en la versión de la antiguinía de aquí, ¿no? Y que va a hablar sobre recursos genéticos en Túnez. Están aquí en el marco de un proyecto ICOP que tenemos entre España y Túnez para recursos genéticos, precisamente. Entonces, pues ya les cedo la palabra y...
Okay. Okay, sorry, I will speak in English, no habla español. Thank you very much for your invitation. Thank you for all the people coming today to listen to this talk that I'm going to give. And I would like especially to thank, of course, Pedro and his team, because we have started collaboration through a DromaMed project. It's a PRIMA project. 4 years ago, this is how the link started. And by that way, also we have a PhD project that was co-supervised of Mohammed Diya, who is currently here, and we are proud to see him, our previous student, working with you, and involved in different projects that you are handling on maize mainly, and other topics. And really it was this day that we spent good interaction with many people of you, and I hope that the presentation I will give will open other possibilities of collaboration, because we will have always different opportunities through EU projects like PRIMA or others to have granted projects, and it's always interesting to have a good network and have collaboration with different parts of the world. So my talk is going to be focused on the use of natural products applied either by seed coating mainly on cereals and root feeding on tomato as an example, to improve plant tolerance against biotic and abiotic stress. First of all, I would like to start to explain a little bit about Tunisian agriculture. So our country is mainly a wheat producer in the north, but is very well known as olive oil producer, as ranked fourth in the world and first exporter, where the olive tree can be observed from north to south with different density, and we can have even one olive trees per hectare in the south, while we have very intensive in the north. And we have also, of course, palm date trees which are exported, and we are first one exporter worldwide of date palm. And Tunisia was known as to be the granary of Rome, so like a supplier of wheat for Rome, but this is a long time ago, because maybe the conditions were so good in order to produce a lot of wheat, and also because it was a Roman colony, so it has to provide the empire, Roman Empire, by wheat. However, today we are really on this main crop, because we use a lot of wheat in our diet, daily diet. We have many problems of production and to supply the needs of the population. We have more or less 1 million hectares. In Tunisia, we have 4 million hectares that could be cropped by different, as I mentioned, crops, but mainly 1.5 million is olive trees and 1.5 million is cereal. Among that, 1 million is wheat, but mainly durum wheat, because we eat a lot, and we have a low average of yield, which is not enough for the population needs, which is about 25 million cantons, since each person eats about 250 kilos of durum and bread wheat. So we import like half of our needs, mainly of bread wheat from Europe and other parts of the world. And of course, one of the reasons of this low yield is due to the climate change, especially because of the drought. We have very bad rainfall distribution among the years and among the season, especially, but you know that it's very important to have rainfall during the grain filling. And also we have different disease. We are very well known by septoria leaf blotch. It's a hotspot of septoria leaf blotch, Tunisia, but also there are other diseases that are emerging because of this difference in climate, like mainly the rust, but also soil-borne disease like Fusarium, which are very difficult to control. So what is our main interest in our laboratory? Our laboratory name is cereal breeding and improvement. So we are working on biotic stress, mainly plant pathogenic fungi and soil-borne fungi, as Fusarium and foliar one like septoria leaf blotch and botrytis disease. So these are handled on two model plants. So we have mainly durum wheat and also tomato, as I will show you later on how do we work with that in our lab, and abiotic stress, especially drought, as I mentioned, but also salinity, which is a negative impact on the plants for especially salty soil or irrigation with salty water. So the natural products that we are focused on are mainly beneficial microorganisms or their extract like yeast, fungi or bacteria, plant extracts, mainly essential oils from thymus or rosmarinus, so from medicinal plants, and also we are working on algae, different algae. Each algae has its own characteristics like Eclonia, Laminaria, Ascophyllum. And now currently we are focusing also to work on microalgae, which is now becoming more a fashion of use of such kind of plant natural biostimulant. So the strategy used starts at the beginning by the characterization of these natural products, which could be, as I mentioned, either these plant extracts, essential oil or beneficial microorganisms. So isolation, purification, characterization through different tools, especially molecular ones, but also biochemicals. And later on, we will start high throughput screening or screening in controlled condition, working in growth chamber with hydroponics, either on wheat or on tomato, but also with seed coating, mainly for the wheat, as you can see here. And we can handle this high throughput screening using this hydroponic system. But later on, we go to field assays, first by working in experimental station for both abiotic and biotic stress. And later on, we can have some application, which is helped by some extension network technical center to work with farmers in order to see the efficacy of these products really in the field. So I will start first of all by the first part, which will start by the identification and characterization of this natural product and how we can qualify them as biostimulants. So the biostimulant now become like a fashion worldwide as agriculture inputs, and there are many definitions according to different parts, but the main one is from the EBIC, which is the European Biostimulant Industry Consortium. And we can consider this biostimulant as any natural product which can have any efficiency in the physiological pathway of the plant by improving its comportment against abiotic or biotic stress, but also to enhance its growth and productivity. And among these, we have different, of course, classes like the beneficial microorganism. Here I will point our work with PGPR bacteria like Parabouchulderia fetiformans. Also, we have, as I mentioned, extracts from plants. Here I will show the example of thyme essential oil. We have also beneficial fungi. Here I will point out our experience with Trichoderma species. And also I will show some highlights about our new project working on ketosone as one of the animal extracts to be valorized. So this is Trichoderma, is very well known as to be saprophytic fungi, but is it used as biological or bio, let's say, pesticide, but biostimulant as well. So we have isolated different species, characterized them by molecular tools. We have isolated also beneficial yeast like the Majorisma one, which was firstly reported, and its extracts as having a very good power in inducing defense response. It was an antagonist yeast that was said by chance isolated in the lab because it has proved to be, as you can see here, antagonist against pathogenic fungi like here on Fusarium. And the PGPR bacteria, so we have isolated different bacteria from extremophile plants from the desert and the southern part, and we think that if these bacteria can help the plants to grow in these different parts, or these difficult, parts of the territory, so that they can help to boost the growth in difficult condition linked with drought or heat stress. So these bacteria were then characterized by full genome sequencing thanks to a collaboration with Sainsbury Lab in Norwich in UK. So we have the facility to sequence the whole bacteria, and this helped in the identification, very accurate, of course, of the bacteria species as well as its genus, because now there is a lot of complications, especially in the bacillus bacteria taxa, and full genome sequencing can give really the best idea. So then we do also the annotation of the genome, like you see here, one of the bacteria, Bacillus safensis, and we can have idea about its potential use as PGPR or as plant inducer. Okay, also we have worked on, as I mentioned, the thyme essential oil, and here, of course, the essential oil are different from very variable according to the plant, where you collecting, the time to collecting, the area. So we have done GCMS analysis to characterize the different essential oil extracted, and also from lemongrass. Here is the GCMS characterization of this essential oil, which is very important to handle this product as it should, therefore, and have a good idea about its efficacy and what are the active ingredients involved in this kind of efficacy. Also, we do work with reference product from commercial companies, like the best one known for trichoderma, like here from Coppert, which is the best well-known worldwide bio solution, and Serenade from Bayer as bacillus-containing product, and also Panoramix as a seed coating product from Coppert. So the characterization starts first with this product for the direct effect against this plant pathogenic fungi, because I'm mainly working on biotic stress due to fungi, but also on abiotic stress on, as I mentioned, drought mainly, and sometimes salinity. So we start by in vitro, of course, activities using different concentration of the active ingredient of the extract, and this was done on different plant pathogenic fungi which have different characteristics, like airborne, soil-borne, mycelium, sterile. This is an oomycete, and this is like a yeast-like fungus. This helps to know exactly the activity of the product that you have as a plant extract, or as I mentioned, beneficial microorganism extract. And here we have the capacity to identify the EC50 in ppms. But since this is very high, cannot be used, as you can see here, in applied condition for the direct effect. Therefore, we focus on the indirect effect of the product. So we are not talking anymore of the biocide effect, but we will focus on the possibilities to have this product working as a plant biostimulant through the plant. And first, I will explain how to work on the seed coating on wheat to try to seek for the inducing effect of these products. So what is the seed coating? Seed coating is a promising approach which is consisting of using a product that you will coat and stick on the seed, and this is something very useful, especially for cereal and field crops, because you have the facility to do very easy application for very large area. So the seed coating treatment consists on using the seed first, then a coating agent. For our case, we use tannin extract natural as a polymer that will stick your active ingredient or extract on the seed. So you can use, of course, water as control, but you can use one beneficial like trichoderma, and you can even combine between different beneficial microorganisms like trichoderma from one side and the yeast from the other side to seek for any additive or synergic effect. So the methodology consists on using this seed coating by doing, as I said at the beginning, a screening in the growth chamber by using some assays on Petri dish or hydroponics or in pots. Then we can go to later on to the research station, either for biotic stress, northwest part, for seeking the resistance or tolerance to the plant pathogenic fungi, or in the center or southern part of Tunisia to seek for the abiotic stress, mainly drought or salinity. And this is assessed by using histological, morphophysiological, biochemical, also molecular and agronomic analysis for different traits. So here I will start by showing a short video which will explain the methodology.
Wheat production is considerably affected by climate change and biotic and abiotic stresses. Let me show you how promising it is to apply the approach of seed coating with natural compounds, as thyme essential oil, and the approach of seed coating with beneficial organisms, as the endophytic bacteria Paraburkholderia phytoferments, and how do they induce the seed priming. Thyme essential oil and the coating product were applied progressively to wheat seeds in continuous rotation until complete adhesion. Thyme essential oil compounds weakens the membrane barriers and lowers the turgor pressure of the cell wall, leading to a larger water uptake, which is crucial for the interruption of dormancy. Once penetrated into the seed, thyme oil compounds induce a transient intracellular acidification that stimulates the electrogenic proton pump, which is responsible for cell enlargement and elongation. Subsequently, peroxidases were induced to scavenge reactive oxygen species, as a consequence of the oxidative burst after acidification. Peroxidases, which are also involved in the lignification, render the plant stronger. All these effects results in the promotion of germination and growth, and illustrate the chemical priming induced by thyme essential oil, resulting in a systemic spread of signals from seeds to emerging leaves. As for the endophytic bacteria Paraburkholderia phytoferments, it was applied progressively, with the coating product, to wheat seeds in continuous rotation. This bacteria produce an enzyme, named endopolygalacturonase, that induce cell wall loosening and extensibility, leading to a larger water uptake, and easier bacterial penetration and colonization of seeds. These effects results in an earlier interruption of dormancy, and an enhanced emergence of seedling. Once the bacteria have colonized the emerging seedling, they produce cell wall degrading enzymes, causing a transient stress, that consequently triggers the accumulation of phenolic compounds, to protect the plant against oxidative stress. Besides, they produce the enzyme ACC deaminase that lowers the ethylene level. They also produce the phytohormone auxin, therefore engendering growth promotion. These effects demonstrate the biopriming of seeds, induced by Paraburkholderia phytoferments.
So this is how we can work, either with, as you can see, PGPR bacteria or plant extract. And now I will show you the main results we obtained for abiotic and biotic stress. I will start with drought tolerance because maybe you are more interested in that. So for the wheat tolerance, okay, sorry about that. So I will try to speed up a little bit and show you that we can, through a controlled condition, seek for the water stress and position, and what is coming from the seed coating with the beneficials, as I mentioned, or by the thyme essential oil extract. So we can do moderate or severe or long-term stress, and for that we use different devices like multispec or NDVI or dual-X in order to seek for the physiological parameters at the plant level during this kind of NDVI. And what we can see for the growth promotion compared to the control, that with thyme essential oil we have improved both rooting as well as shooting as well as for the bacteria, and this is mainly due to all the physiological parameters that were collected as well as the discrimination of the nitrogen and carbon isotope showed that there is an enhanced photosynthesis with higher ABA accumulation from one side and downregulation from the other side, and that the accumulation of flavonoids and anthocyanins from one side while soluble sugar and flavonoid from the other side, and this is enhancing the carbon metabolism for the plants. This is also showed that under the water nutrient stress we have a better efficient status due to all these devices that have been used in order to seek for the physiology of the plant, but also balance the nitrogen carbon metabolism, while compared to the control for both the induction of elongation as well the induction of lateral root development with the bacteria, and for both we have lower ABA content where there is an early peak of ABA for the other one, and as I mentioned also this was assessed by the carbon isotope discrimination and showed high photosynthesis and phytoprotective process. Under field conditions this was done in semi-arid area by using drip irrigation with 100% or 50% supplemented the plants, and as the same as mentioned before, we have used different devices to control the physiology of the plant and the yield component at the harvest. This showed that for the growth and yield component on 2 different seasons we have an improvement, while we would do work without stress with both an improvement of the yield by using the thyme essential oil or the bacteria, but mainly when we go to the stressed condition we can see that there is an improvement of the yield in stressed condition with 50% here, as you can see, irrigation, and the effects on weed physiology showed to be at the grain filling at the heading, grain filling at the harvest stage, and this is due to higher leaf content of carbon, of isotopic carbon, as well as higher stomatal opening and lower canopy temperature, which result in higher straw and grain yield and improvement of the southern kernel weight. So for the bacterial strain, it have contributed to higher content of carbon, and as you know, for carbon discrimination, the more the plant discriminate, the more the plant breathes. So the more there is discrimination, better is the status of the plant. With enhanced nitrogen and carbon assimilation, while for the thyme essential oil there is a lower leaf total carbon with enhanced carbon mobilization and lower straw nitrogen content but enhanced nitrogen mobilization. So for the water stress, if we compare what are the mechanisms that have been used by the bacteria, it is clear that the bacteria will contribute more to the plant investment into the grain formation and also later on the grain filling, while the thyme essential oil is helping more the plants for adaptation efficiency and an impact which is centered on either vegetative growth, tillering, or tiller fertility depending on the climatic condition. So now I will switch to another example, which is the biotic stress, which is now for the septoria protection, which is, as I mentioned, one of the main fungal pathogen of wheat in Tunisia. So for that we have done an assay in controlled condition, and we have used the coated seeds. Then we did at 3-level stage an inoculation with the spores of the septoria. Then we did the analysis using histochemical and microscopic observation, as well as the biochemical analysis, and did an assessment of the necrosis using MBG software. And if we compare between the control, where we have rare papilla formation, and we have vegetative growth of more growth, sorry, of the mycelia with necrotrophic area, which has high as well the pycnidial density, which is showing the necrotrophic and biotrophic phase of the pathogen. There is an accumulation of hydrogen peroxide as well as peroxidized catalase and phenolic compounds, while where we see and observe the plants which are inoculated with the pathogen but which are coming from coated seeds with thyme essential oil, we have reduction of the systemic growth with some cell death, which will block and stop the invasion of the pathogen, and more papilla accumulation. The papilla is a very important signal that shows that the plant is defending against the pathogen, but also for the necrotrophic area as well as the pycnidial area, we have, of course, less area which is covered, which is due to lower hydrogen peroxide, but more peroxidase and phenolic compound. Finally, we have the same thing observed with cell death as well as less colonization of the pathogen and more papilla formation with less necrotrophic area for the seed coated with the beneficial bacteria with lower hydrogen peroxide and also catalase, peroxidase and phenolic compound. So the thyme essential oil will contribute more to systemic acquired resistance, which help to the plant to overcome the pathogen invasion, while the bacteria will work on what we call induced systemic resistance, which help the plant, of course, to overcome the pathogen. So I think because of the time running, I will just explain how the mechanism is involved in the septorial leaf blotch. So we have decreased chlorophyll in the control, and there is an increase of stomatal opening with decrease of canopy temperature, while with the beneficial microorganism and the plant extract, we have higher chlorophyll and we have lower stomatal opening and lower canopy temperature and higher chlorophyll in the case also of the thyme essential oil with lowest stomatal opening, and these two will help to have higher carbon content in the grain, and will have more discriminant carbon as well as nitrogen. And as I mentioned, this is something which helps to understand that the plants are overcoming the stress due to that. So we have done also with seed coating for Fusarium root rot. I will skip this because of the time running, and this was done by coating with the trichoderma alone or with the yeast alone or the combination of both. And also we have observed the phenomenon shown before that we have a priming effect. That means that we have induced of the biochemical markers linked to the defense response by lower H2O2 and more peroxidase, and enhanced of phenolic compounds which are involved in cell wall strengthening, and also increase of the chitinase, which is known as a peer protein that helps to defend the plant against fungal pathogens which are rich in chitin in their cell wall. So we have more catalase, more peroxidase, so strengthening cell wall, less hydrogen peroxide, so less stress, and more chitinase activity, which help to degrade the pathogen. So to explain, when we have attack with the Fusarium, with the pathogen, this is help to have what we call inducing or elicitation effect by inducing these PR proteins into the plants. And on the other side, when you have the beneficial and the extract of the trichoderma, which contain also chitin-like oligosaccharides, which will work as elicitors, we have an induction of the defense response. So when you have both at the same time, the pathogen as well as the beneficial, you have a priming stage which will help to have more efficacy of the defense response of the plant. So I will skip this about the Fusarium head blight because of the time running. So what I would to say that in the efficacy of the seed coating affects plant physiology at all stages of development, recorded from the seed germination until harvest, and it can help for local as for Fusarium by the roots local defense, inducing local defense, but also systemic acquired resistance, which goes from the roots up to the leaves and helps, for example, to overcome the problem to septoria. So I will skip this because of the time, but we are working also on root feeding on tomato by hydroponics, and we are testing the same thing for the beneficial and look for either without inoculation what helps to boost the growth of the plant or to reduce the infection due to Fusarium, Fusarium oxysporum radicis, which is used quickly here to inoculate the plant, as you can see here, by the root feeding, as well as the inoculation of the leaves by the botrytis, which is an airborne pathogen, to look for the systemic acquired resistance, and this is done in controlled condition and followed by also biochemical and molecular analysis, which shown all together. I will skip this to show you that we can look for and see an efficiency of this application on the roots by root feeding of the beneficial, because it helps for systemic acquired resistance as well and limit the infection due to the foliar pathogen, but also for local resistance by induction of the resistance at the root level. So just to finish, what is the priority in the plant? Here I show an example for the Ukrainian war, for example, where it is known to be a very good producer of wheat, but when you have war, what is the priority? The priority, of course, is to defend. So the plant, when it is in a bad condition, will go for defense, which is a priority, rather than a production. But the plant, when it goes for the defense, it will take from its energy upon the production. So the best thing is that the plant will go through a balanced situation between protection and production in order to be able to protect the plant, but also to ensure production. And for that, I would like to say that the biostimulant usually work on different pathways, which are well known as jasmonic acid, salicylic acid, or IBA. And the best thing is when you use biostimulant in order to have better efficacy into the plant response, that they act on different pathways. So therefore you can have an additive effect or a synergic effect into the plant, which will help to overcome different stresses, which could be abiotic or biotic. And just to finish quickly, I would like to say that for all this work we do, also in addition to the work as academic and at the research station or at the lab, we do also the work at the field station, at the field level, with farmers, with seed coating, for example. We have convinced some farmers to do the application of the biostimulant by seed coating, in addition to the treatment by fungicide that they do. And we have seen that this helped to improve the yield, as you can see here, when we do the biostimulant itself or the biostimulant in combination with the fungicide. Also, we did that for potato because we have a collaboration with Potato Technical Center, and because of the problem of using sliced tubers, because of the higher price and of the seeds for the farmers, for the local farmers, they go to make sliced tubers, and this, of course, is a problem to open for the infection by different pathogens. That's why we helped them by having another solution, which is using the seed coating, the sliced tubers by coating, in order to overcome the problem of infection at the different level, either for soil-borne pathogen like rhizoctonia or airborne pathogen. And this is like an extension activity, but also we do that for tomato with nurseries as well as with farmers, to show how we can have efficacy by using biostimulant to improve the plant growth, but also the quality of the tomato, for example, by analysis of some parameter linked to the quality. And as I told you, now we are working with a startup in Tunisia to valorize the ketozone, which is coming from blue crab. Blue crab is a new invasive species for fishermen in Tunisia and is making a threat, and now is valorized because the extract of these crabs are used to make ketosone, and ketosone is very well known biostimulant that helps to overcome abiotic and biotic stress. And currently we are doing this experiment both on potato and wheat, and we have very nice and good and encouraging results, but we are still analyzing that. So finally, I would like to thank the collaborator with who we worked different years, mainly Professor Jose Luis Arauz for all the physiological analysis and who is involved with us in different PRIMA project, but also groups from University of France, the French team, and from Italian, and also collaborator from the private sector. We have had different agreement with SAS-CODA from Lerida, for example, is a Spanish company making biostimulant, and also Alga Energy, Silva team from Italy, Coppert from Netherlands, Simborg from Alicante in Spain, and we worked with them in order to prove the efficacy of their product and explain the mechanism of action. This work has been done thanks to different PhD students and master students that I would like to thank them for their efforts that they made to have all these results. And finally, I would like to thank you for your attention, and I would like to mention that National Agronomic Institute in Tunisia is the oldest agronomic institute in Africa, and now is celebrating not 120 and I think 128 years old. Thank you very much for your attention, and I'm sorry if I was a bit long.
Hello, good morning everyone. I am Zoubeir Chamekh from the National Institute of Agriculture Research at the National Institute of Agriculture Research in Tunisia, created in 1930 before it's named École Coloniale d'Agriculture. This is during the French occupation. In Iraq, staff have about 73 scientists and 54 technicians and more than 235 labor. We have in Iraq 7 laboratories, field crops laboratory, horticulture, plant protection, biotechnology, animal and forage production, agronomy, science and technology, and the last one is the rural economy, where there is the field. I belong from the field crops laboratory. We have also the 5 experimental station. I belong to the field crop laboratory. My research focus is to create a new variety in context of climate change. Okay, Tunisia located between Algeria and Tunisia, and we have about 165 square kilometers and a population more than 12 million people, with a total area, with a total arable land about 4.5 million hectare, during wheat production is heavily dependent. Most of the wheat production is growing in the center of Tunisia, characterized. This is the arid area, characterized by rainfall. It's less than 115 millimeters. Wheat is the most important crop Tunisian, both for cultivation and consumption. This figure shows the importance to improve wheat production. For the cereal in Tunisia, the main limitation production is it drought, the first one, followed by disease and the inappropriate agronomic practice. Wheat production wheat production in Tunisia, it is very related to rainfall, and we need to improve production for the people. The main disease weight in Tunisia, it is septoria. It has traditionally been the most important wheat disease, but however, due to the climate change, now rust becoming the most important disease affecting wheat production. We have the yellow rust, stem rust, and the leaf rust. The main objective of cereal breeding program in Tunisia is to create a new variety with high yield, good technological quality, and tolerant to different abiotic and biotic stress. To do this, we create a maximum of genetic diversity by hybridization, make to cross, and introduce of segregation line from Semite and Icarda in order to create this genetic diversity. This figure summarizes the different stages of the breeding process from crossing, here, crossing block, to release, to registration of the new line. The crossing block was continuously enriched with germplasm from Semite, and we start the selection from F2 to F5 in order to select the best line for disease tolerance, agronomic type, precocity, and large adaptation. Starting at F7, drought stress trial and unused drought stress trial and unused quality were tested before to go in the different to assess this line in different station. Okay, in our program we adapt a strategy as named genetic pyramiding in order to put the maximum of gene related to disease tolerance in the same genotype, because rust menace continued fate with any old enemy, because we have a lot of collaboration with the Semite and the Icarda, and we received every year a lot of material characterized, and all the gene was characterized in this introduced line. This is the case of the stem rust, this is the strip rust, and this is the leaf rust. Now selection for drought tolerance, we started from F5 until F7. And our thinking is to skip the segregation population for drought tolerance, hoping for a high frequency of advanced line performance in drought stress. Here we show the first year yield trial. We have about 200 entry plus check, and the second year we have, after selection, we have 100 entry, and the third year, this is the advanced yield trial, we have only 50 entry plus a check. We have tested this in the drought tolerance phenotyping platform in the center of Tunisia, and the heat tolerance at the KEF station. After this, results will be analyzed, and the best line will be selected for their yield production. Here we show the distribution of different line in the control condition. Here compared with the check, and here under stress condition compared also with a check, and we select the best line for the advanced yield trial. This line will be analyzed the quality testing before to go in the registration. This is we analyze the 1,000 kernel weight, grain propriety, milling propriety, flour propriety, gluten also, and bread making quality. Given that the zinc is an essential macro element for health, and genotypic variation in grain zinc concentration has been demonstrated in wheat, like this. This is the microscope, electronic microscope. We make also evaluate the zinc content of the different advanced line before to propose the inscription to registration and select the best line with the high zinc concentration. And we go to analyze the distribution of the different group of line and their origin. For example, this is from origin from Tunisia. This is the third group from Semite. This group from Icarda, and this group for the Landraces variety. Under field trial, we make also different traits, measured different traits like ion concentration, carbon isotope composition, biomass, plant height, kernel thousand weight, and kernel number of kernel per meter square, in order to select the best criteria used for selection. And as you know, the carbon stable isotope composition, it is very assessing to tolerance to drought. Also, we have measured leaf water content, leaf potassium, leaf calcium content, and leaf sodium content. The main achievement in the cereal program, breeding program in Tunisia, it is to register many varieties from bread wheat, durum wheat, barley also, and this is the last obtention of bread wheat. In addition, we participate also in different development and extension projects addressed to farmers in order to improve their resilience by implementation of different innovative solutions, like agronomic sequence, use the line tolerant to salt stress, here, use the forage and introduce the forage species, and introduce also the early vigor line. Different innovative approach was used, like participatory workshop, on-farm experimentation, and the multi-stakeholder platform. We also implemented different living lab in different places in Tunisia in order to share this innovative solution to farmers. This is a multi-stakeholder platform. We also contribute to capacity building in order to develop farmer skills. our strategy is based to choose the leader farmers where implemented the experiment, and he shared with their neighbor this innovative system. Also, we have installed a training platform where here implemented a different system of irrigation and different variety, different protocol to fertilizer for farmers and extension service of the ministry, student and the stakeholder. Thank you for your attention.